Hello everybody. This mini lecture is brought to you by Mrs. Draves, and I am going to be teaching you about some of the random processes that occur in nature over time that lead to evolution. And evolution can occur through random processes as listed here. The first one that we're going to go over today is mutation, and then we will go over what gene flow is, followed by genetic drift, then bottleneck effect, and finally the founder effect. All of these processes here occur randomly, and there are some exceptions to that though because of the influence of the anthropogenic effect of humans, and we'll get into that a little bit later. But most of these are random events that lead to population change over time. The first one I want to go over is mutation. And I'm sure you've heard of this before, especially in biology class, but a mutation is a change in the genetic frequencies of a population over time. It's actually a change in the genes over time and the relative percentages and ratios of those. And a perfect example of this is the rock pocket mouse, which lives in the southwestern United States, and we'll actually do a lab on this, where a mutation happens in a population. So for example, in this original population of mice, 90% of the population was white and 10% was black. The mouse that's black is not a different species, it's actually the same species, but it actually has a mutation in a protein, and so as a result it has a black color. So that mutation occurred in that population, but over time it was not lost. Over time it actually increased in frequency. So you could see here as multiple generations went on through time, this population went from a 10% black to 30% black. So this is called a mutation, and it changes the frequency of phenotypes or physical traits that you see over time. In the lab, we will get into greater detail with that. The second type of random process is called gene flow. And gene flow occurs when individuals move from one population to another population. And so what happens is the population that they left changes in genetic composition. And then the population that they're going to changes as well because of their presence. So the evolution by gene flow, a perfect example of this that includes human influence and conservation biology, quite honestly, is the Florida panther. So the Florida panther is actually an endangered species, and its population greatly declined in numbers. And whenever there's a decrease in numbers, okay, there is a decrease in diversity or variation. So decrease in diversity or variation of genes. There's low genetic variation. Okay. Now, when that happens, inbreeding can occur where individuals will mate with individuals that they're somewhat related to, and so as a result, mutations occur to where different types of physical abnormalities can happen. So, for example, in the case of the Florida panther, they showed signs of inbreeding by number one, they started to have kinky tails, kind of like tails that were like a pig. They also saw that through analysis that their hearts had defects in them. And they also had low sperm counts. So these are three phenotypes that were expressed as a result of low genetic variation. So what conservation biologists did was they decided to manage this situation by introducing 8 panthers from Texas. And so, as you can see here over time, this is your population here. Introduce a few individuals from Texas, and what that did was that brought in some genetic diversity. And their population has grown since this introduction from 30 individuals to 160 individuals. And this was published in 2015. So, it may be a little bit different today, but probably not by much. So it's also important to know that when you have individuals from one population going to another population, that does occur naturally, for sure. But in this particular example that I gave you, we kind of put a little conservation biology twist on it. The next one is genetic drift. And genetic drift occurs, as you can see here, in this small population of mice. Some less common genotypes can be lost by chance, and that's just because of random mating. Because it's such a small number of individuals, it can just be lost by chance. And that's a really important thing to remember, is chance, because a lot of it's by chance. The less common genotype then does not become part of the gene pool and doesn't mate. So it's not successful. So as a result, the genetic composition can change over time, as you can see here. So it just goes from 40-60 to 20-80 to 0-100. So you see this drift in relative percentages of phenotypes over time. Now in a large population, it's much more difficult for less common genotypes to be lost because the populations are large. So as a result, the genetic composition tends to remain somewhat stable over time in large populations because a number of these individuals is large, and so they have this absolute number of large individuals. There's a greater chance of them mating, and so there's a greater chance of that phenotype being persistent through the population. So it's important to know that genetic drift is more visible, I guess you could say, more evident in smaller populations. The next one is the bottleneck effect. And so I really want you to think about a bottleneck. That's why they call it like this. Kind of like an Erlenmeyer flask. And you start out with this population years ago, let's say. And the population exhibited lots of diversity at that time. So the diversity is evident by all these different colors. Okay? And even the length of these lines. Okay? So that you can consider this like beak size, right? Beak length, beak thickness, that kind of thing. So there's lots of diversity. And then all of a sudden, that population is reduced. And it's reduced through, it could be reduced through a disease coming through. It could be reduced through a natural disaster. It could be reduced through poaching. So there's natural ways for it to be reduced as well as anthropogenic sources. And so what that means then, this part of this bottle is that pressure, that reduction. So then what you're left with, if we were to pour this out because of that huge reduction in population, is maybe something like this. And that's it. So all the other variations that you see in this original population and genetic diversity become reduced to this. So this low genetic variation in a population is not a good thing, and that's because there's definitely an increase in the risk of disease and there's low fertility. If there's environmental change, which there always is, they are less apt to adapting to that change over time, and eventually it could lead to the decline and extinction of the actual species. So bottleneck effects, you could pretty much safely conclude that all endangered species or species of concern have had some type of bottleneck through, in most cases, human interaction. So it could be habitat loss, it could be climate change, it could be poaching. Any of those things, and that drastically reduces the size of the population, which leads to further change in that same direction, which we all know is a positive feedback loop. Okay? So an example of this is actually the northern elephant seal. And the northern elephant seal had a huge bottleneck event in the late 1800s, which we actually find this time to be one where there's lots of huge population declines due to over harvesting. And so as a result of human hunting in the late 1800s, the population decreased to about 20 seals. Now we have about 30,000 seals. But the problem is that original 20 population that we started with, that we tried to conserve and work with different types of reproductive methods to bring their population numbers up, it wasn't that diverse. So there was a huge reduction in the variation of this species. And so that's a huge concern. And so there's different things that different zoos and aquaria are doing to intervene with this, and that's all in with reproductive biology and conservation biology. You could also consider it like wildlife management. And then the last one I want to talk to you about is the founder effect. And this basically is a change in the genetic composition of a population as a result of descending from a small number of colonizing individuals. So basically you have a mainland, and this mainland has all of this diversity here. And so a few individuals decide that they want to fly to another area, and they by chance come across an island, and they stay there and they populate there, they colonize there and populate there. And so that founding population is pretty much like the original population. So the genotypes on the island will represent only a subset of that original population right here. So these individuals here, this population is only a subset of that mainland population. And as with the bottleneck effect, some of these genotypes will not be present in the new population. Okay? So that is the founder effect. So that concludes this lecture. All of these events that I just described to you all are components of evolution and how it is a random process. The end.